Hello students, welcome to EPG Pathshala. I am Professor Ramesh Kothari from Saurashtra University, Rajkot, Gujarat. Today we are going to discuss about a module on pentose phosphate pathway under paper carbohydrate metabolism. So students, let us see what we are going to learn in this module. Students, we will try to understand the function of pentose phosphate pathway in production of NADPH and ribose, precursor for synthesis of nucleic acid. We will also try to understand the importance of NADPH in protection of cell against highly reactive oxygen species, and we will also try to relate defect in pentose phosphate pathway to disease condition. The pentose phosphate pathway is the major source for NADPH required for anabolic process. There are three distinct phases, each of which has a distinct outcome. Depending on need of organisms, the metabolites of that outcome can be fed into many other pathways. Glutathione is directly connected to pentose phosphate pathway. As the need for glucose 6-phosphate, the beginning metabolite in the pentose phosphate pathway, increase, so does the activity of gluconeogenesis. The main molecule in the body that make anabolic processes possible is NADPH. Because of the structure of this molecule, it is readily donates hydrogen ion to metabolites, thus reducing them and making them available for energy harvest at a later time. The pentose phosphate pathway is the main source of synthesis for NADPH. The pentose phosphate pathway is also responsible for production of ribose 5-phosphate, which is an important part of nucleic acid. Finally, the pentose phosphate pathway can also be used to produce glyceraldehyde 3-phosphate, which can then be fed in the TCA cycle and electron transport chain cycle, allowing for the harvest of energy. Depending on the need of the cell, certain enzymes can be regulated and thus increasing or decreasing the production of desired metabolites. The enzyme responsible for catalyzing the steps of pentose phosphate pathway are found most abundantly in the liver, the major site of gluconeogenesis, more specifically in cytosol. The cytosol is where fatty acid synthesis takes place, which is a NADPH dependent process. The figure shows the concept map of pentose phosphate pathway. Pentose phosphate pathway has two parts, that is oxidative phase and non-oxidative phase. Pentose phosphate pathway protects cell against reactive oxygen species. Regulation of PPP is an important point. An importance of PPP, pentose phosphate pathway, for the cell protection is also an important aspect. Glucose is catabolized via glycolytic pathways into two molecules of pyruvate, and then this pyruvate is oxidized via citric acid cycle to generate ATP. In animal tissue, there is another metabolic pathway which is called pentose phosphate pathway, which is also known as hexose monophosphate pathway, that is HMP shunt, or phosphogluconate pathway. The oxidation of glucose without direct consumption occurs by pentose phosphate pathway. In mammals, NADPH acts as universal reducing agent in anabolic pathway. Mammary gland adipose tissue, liver require large amount of NADPH for synthesis of fatty acid and glycerol. Tissues less active in fatty acid synthesis, such as a skeletal muscle, lack this pathway. The generation of pentose, that is ribose sugar, is necessary for synthesis of nucleic acid. Erythrose for phosphate generated by pentose phosphate pathway is utilized for the synthesis of aromatic amino acids. Reactions of pentose phosphate pathway occur in the cytosol in two phases, as NADP plus is used as hydrogen acceptor. The pentose phosphate pathway can be divided into two phases. Phase 1 is called irreversible oxidative phase, and phase 2 is reversible non-oxidative phase. During first step, glucose 6-phosphate is oxidized into ribulose 5-phosphate and carbon dioxide. During oxidation of glucose 6-phosphate, NADP plus is reduced into NADP. During second step of pathway, ribulose 5-phosphate converted into other pentose 5-phosphate, including ribose 5-phosphate, used to synthesize nucleic acids. During third step, a series of reactions occurs which convert 3 of pentose 5-phosphate into 2 molecules of hexose and 1 molecule of triose. During fourth step, some of these sugars are converted into glucose 6-phosphate, so the cycle can be repeated. Here, oxidation reaction of C1 position is occur. If you see in the figure, hydride transferred to the NADP plus, forming a lactone, which is an intramolecular ester. Reaction start with glucose 6-phosphate dehydrogenase, which oxidize the aldehyde at carbon 1 position of glucose 6-phosphate to a carboxylate group. Glucose aldehyde normally exits as a hemiacetal pyrene ring in which aldehyde has combined with O5. The carboxylate anion driven from glucose is gluconate, and cyclic ester of this type are called lactones. Hence, the product name is 6-phosphogluconate lactone. This is a very favorable reaction in forward direction for producing NADH. The reaction shows the action of enzyme lactonase. The lactonase opened by hydrolysis the additional water molecule to cleave a bond, usually a type of amide or ester. In this case, since lactone is intramolecular, then a 6-phosphogluconolactone is opened up to the acid forms gluconate. This type of hydrolysis reaction is favorable. Following reaction shows the action of enzyme 6-phosphogluconate dehydrogenase. 6-phosphogluconate dehydrogenase carry out the conversion of a 6-carbon skeleton to a pentose with oxidative carboxylation. The C1 carboxylate is removed as the C3 position is oxidized to a ketone, making 5-carbon ketose, that is ribulose. This is an example of acid-base catalyzed reaction. Ribose 5-phosphate has no immediate use in the cell, so is rearranged into other form isomerization of ribose 5-phosphate, which can be used for RNA, DNA and nucleotide. non-oxidative phase of pentose phosphate pathway. It starts with recycling pentose phosphate to glucose 6-phosphate. In cell that require high level of NADPH for biosynthetic reactions, the ribulose 5-phosphate produced in the oxidative phase need to be converted back into a glucose 6-phosphate to maintain flux through the glucose 6-phosphate dehydrogenase reaction. The carbon shuffle reaction of a non-oxidative phase, which ultimately are used to generate glucose 6-phosphate using trans-ketolase and trans-aldolase enzyme. The figure shows the action of enzyme ribulose 5-phosphate epimerase. Epimerization reaction is in turn of a stereocenter in the substrate. An epimer of a compound occurs when only one stereocenter is flipped. Phosphopentose epimerase catalyzes the synthesis of xylulose of 5-phosphate from ribulose 5-phosphate, and the reaction is bidirectional. Figure shows the trans-ketolase catalyzed reaction. Trans-ketolase enzyme use cofactor called TPP, that is thiamine pyrophosphate. Thiamine pyrophosphate is used to stabilize a 2-carbon ion intermediate. The following figure shows the reaction catalyzed by trans-aldolase. Trans-aldolase forms a protonated skip-base intermediate with a ketose stabilizing 3-carbon intermediate, allowing an aldehyde base sugar to react with enzyme-linked ketose. Following figure shows reactions required to convert 5-pentose to 6-hexose. The pentose are converted into 6 and 3-carbon sugars. The way to decipher it is to remember 2 key concepts. First is either 3-carbon unit, that is 1 reaction, or 2-carbon unit, 2 reaction, are transferred between acceptor and donor molecule. The enzyme responsible for 3-carbon transfer is called trans-aldolase, and the enzyme responsible for transfer of 2-carbon units are called trans-ketolase. And second is the number of carbon involved in the reaction add up to either 10, that is 2 reaction, or 9, that is 1 reaction. Figure shows the conversion of 5-pentose to 6-hexose. Trans-ketolase transfer a 2-carbon fragment containing ketone group from xylulose 5-phosphate to ribose 5-phosphate. Trans-ketolase require TPP, derivatives of vitamin B1 as coenzymes, and magnesium ion as a cofactor. Transfer of 2-carbon fragment to 5-carbon ribulose 5-phosphate yield pseudohaptulose 7-phosphate and glyceraldehyde 3-phosphate. The following figure shows the reaction required to convert 5-pentoses to 6-hexoses. Again, trans-aldolase catalyze transfer of 3-carbon from pseudohaptulose 7-phosphate to glyceraldehyde 3-phosphate to form erythrose 4-phosphate and fructose 6-phosphate. Following figure shows the reaction required to convert 5-pentose to 6-hexose. Again, trans-aldolase catalyze a transfer of 3-carbon from pseudohaptulose 7 to glyceraldehyde 3-phosphate to form erythrose 4-phosphate and fructose 6-phosphate. There are 2 main classes of enzymes responsible for the rearrangement and synthesis of different length carbon chain molecule. These are transketolase and transaldolase. Transketolase is responsible for the cleaving of a 2 carbon unit from glycose 5-phosphate and adding that 2 carbon unit to ribose 5-phosphate, thus resulting in glyceraldehyde 3-phosphate and pseudoheptulose 7-phosphate. Transketolase is also responsible for the cleaving of a 2 carbon unit from glycose 5-phosphate and adding that 2 carbon unit to erythrose 4-phosphate, resulting in glyceraldehyde 3-phosphate and fructose 6-phosphate. Transaldolase is responsible for cleaving the 3 carbon unit from pseudoheptulose 7-phosphate and adding that 3 carbon unit to glyceraldehyde 3-phosphate, thus resulting in erythrose 4-phosphate and fructose 6-phosphate. The end result of the rearrangement phase is a variety of different length sugars which can be fed into many other metabolic processes. For example, fructose 6-phosphate is a key intermediate of glycolysis as well as glyceraldehyde 3-phosphate. Pentose phosphate pathway protect cell against reactive oxygen species. Role of NADPH and glutathione is protecting cell against reactive oxygen species. RBCs lack mitochondria and thus the lack of enzyme of citric acid cycle. Therefore, glucose is metabolized exclusively by glycolytic cycle, that is 90%, and pentose phosphate pathway, that is 10%. The pentose phosphate pathway is also responsible for maintaining high level of NADPH in red blood cells, which is used as a reductant in the glutathione reductase reaction. Glutathione is a tripeptide that has a free sulfhydryl group which function as an electron donor in a variety of coupled redox reaction in the cell. In erythrocyte, electrons from glutathione are used to keep cysteine residue in hemoglobin in the reduced state and for reducing harmful reactive to oxygen species and hydroxyl free radical that damage protein and lipid through oxidation reduced cleavage reaction. Figure shows the role of glutathione reductase in flavoprotein that contains the coenzyme FAD and is related to ferredoxin NADP plus reductase. To maintain the reduced state of glutathione reductase use 2 electrons available from NADPH. Figure shows the role of NADPH and glutathione in protecting cells against reactive oxygen species. In erythrocyte, to reduce hydrogen peroxide level through a GSH-dependent redox reaction catalyzed by the enzyme glutathione peroxidase, high level of GSH and therefore high level of NADPH are needed. When erythrocytes are exposed to chemicals that generate high level of superoxide radicals, to reduce this damaging compound, GSH require in erythrocyte sufficient level of NADPH to maintain the GSH-GSSG ratio at about 500 to 1 normally provide an active response phosphate pathway. Role of NADPH and glutathione in protecting cell against highly reactive oxygen derivatives as discussed reduce the glutathione. Flavism was also caused by the same enzymes defect. It was observed that certain people eat food containing fava beans, a main ingredient in the Mediterranean dish falafel. They would become very sick. It is now known that same acute hemolytic anemia seen in individuals with G6PD who treated with primequine also explain symptoms of favism. One of the active compound in fava bean is called vicine, a toxic glycoside that induces oxidative stress in erythrocyte. The following figure shows the regulation of PPP. The first step of pentose phosphate pathway is irreversible committed step. This reaction is catalyzed by glucose 6-phosphate dehydrogenase. This step is allosterically regulated. NADPH, the product of this reaction, is strong inhibitor of glucose 6-phosphate dehydrogenase. So when the cytosol concentration of NADPH is high, the enzyme activity is low. As NADPH is useful in various pathways, inhibition is relieved and the enzyme is accelerated to produce more NADPH. It also allosterically regulated by fatty acid acyl transferase acyl ester of coenzyme A. The transcription of the gene for this enzyme is under hormonal regulation. The synthesis of glucose 6-phosphate dehydrogenase is induced by the increased inulin or glucan ratio after high carbohydrate meal. Insulin, which secrete in response to hyperglycemia, induce the synthesis of glucose 6-phosphate dehydrogenase and 6-phosphogluconate dehydrogenase, increasing the rate of glucose oxidation by pentose phosphate pathway. The synthesis of glucose 6-phosphate dehydrogenase is repressed during fasting. Importance of pentose phosphate pathway. NADPH is needed for reductive biosynthesis of fatty acid, cholesterol, steroid hormone, and sphingosine. Thus, it is active in lectin mammal gland, liver, gonal adipose tissue, and adrenal cortex. NADPH is used in hydroxylation reaction during metabolism of phenylalanine and tryptophan. NADPH is used for the synthesis of nitric oxide. Nitric oxide is a laughing gas used as anesthetic, cause relaxation of vascular smooth muscle. In macrophage, nitrous oxide is effective against viral, fungal, protozoal infection. Nitrous oxide potent inhibitors of platelet aggregation, and nitric oxide also act as a neurotransmitter in brain. Following table shows the summary of pentose phosphate pathway. As we see that there are two stages of pentose phosphate pathway, that is oxidative phase and non-oxidative phase. The enzymes of oxidative phase are glucose 6 phosphoglucoronase and phosglucolactonase and 6-phosphoglucolactonase dehydrogenase. And in non-oxidative phase, enzymes are trans-ketolase and trans-aldolase. So students, let us summarize what we have learned in this module. The pentose phosphate pathway primarily generate NADPH, ribulose 5-phosphate, fructose 6-phosphate, and glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate can be shunted to glycolysis and oxidized to pyruvate. Alternatively, glyceraldehyde 3-phosphate can be utilized by the gluconeogenic enzymes to generate more 6-carbon sugar, that is fructose 6-phosphate or glucose 6-phosphate. NADPH is employed in a reductive reaction in anabolism, particularly in the synthesis of fatty acids. In red blood cell, for example, the major role of NADPH is to reduce the disulfide form of glutathione to the sulfidyl form. Reduced glutathione is important for maintenance of the normal structure of erythrocyte and for maintaining hemoglobin in the ferrous state. Deficiency of oxidative pathway enzyme glucose 6-phosphate dehydrogenase cause hematological problem because the inability to maintain reduced glutathione results in accumulation of damaging peroxide. Glucose 6-phosphate deficiency is associated, however, with resistance to malaria parasite Plasmodium falciparum because the weakened red blood cell membrane, that is erythrocytic host cell, cannot sustain the parasitic life cycle long enough for productive growth. So this is all about pentose phosphate pathway. Thank you.